Anybody have any questions? Yes.
How does the actual bacteria differentiate between its physical DNA and a plasmid? How does it differentiate? Are they treated the same?
Treated the same how?
Does it read both the same? I don't know. How in which way is it just the form of the plasmid that's different and the way it can actually spread around between cells is different?
Yeah, okay. So in some cases, plasmids can actually, like you'll learn about this eventually, but with plasmids, they can actually incorporate into the genome and actually come out as well. But there's like certain type of plasmids that'll do that. Also, there's a plasmid called the F plasmid, and that'll actually, bacteria can share that between them without actually dividing. But they'll read the DNA using the same machinery. So they'll make it into RNA, and then the RNA will be made into protein. So you could have a gene for okay, so antibiotic resistance, you could have antibiotics resistance like in the genome, but with the bacteria that you work with, there are a strain that actually has recombinase and certain enzymes knocked out of its genome, so it can't actually incorporate plasmids into their genome and stuff. But that's kind of more detailed, and I don't want to confuse too many people. If that makes sense.
So plasmids are like DNA outside of the nucleus?
Well, okay, the thing you brought that up, okay.
Do you know if bacteria have nucleus?
Yeah.
Oh no, they don't.
Exactly.
Right.
Oh, okay. So do we have plasmids in our cells, or is that just bacteria alone?
No, bacteria and archaea. Wait, that part might be prokaryotic cells have plasmids.
Oh.
Eukaryotic cells don't.
So is the plasmid kind of something that feeds off the host cell in a way then, kind of?
Yes, that's actually a really good question. So remember how we talked about putting an antibiotic resistance on the plasmid? So that the cell actually keeps the plasmid, right? Because if you don't use antibiotic selection, what the cell will do is it'll actually dump the plasmid because it doesn't need to make it, and it costs energy to run these machines that duplicate your DNA. And if you don't need to make this DNA, why would you want to?
Yeah.
Right? So it is energetically costly, but the bacteria will keep it around because of that resistance, because without it, it would die, right?
So it's selected for.
Sorry?
It's like selected for.
Exactly, yeah.
Maybe I should.
Any more questions? Yeah.
Would it be possible to work with eukaryotic cells? Would you have to sort of cut the plasmid like you mentioned so that it becomes linear, or would that not work?
Yeah, iGEM actually has a few different, what they call chassis, which are essentially the type of cell you work with, or the type of organism. And the thing with working with eukaryotic cells, because they do work with yeast, as well as a worm.
C. elegans and
Yeah, C. elegans. And those are eukaryotic organisms. So they do have biobricks for them, but the thing is, how do I get into this without going too much? Okay, so if you go back to, okay, so if you look here, when Jenna was talking about the universal code earlier, so this is how DNA is read and turned into protein. So these are three-letter codes, and if you notice, a lot of these amino acids, which make up proteins, are coded for by multiple codons. Codons are the three-letter codes, three-letter genetic code. So certain organisms will actually prefer certain codons coding for their amino acids because of what's called, well, because of the carrier that brings the amino acid to the ribosome, which is the machine that makes the protein. So certain organisms will have preference for certain codons for their amino acids because they have the carriers that match up with these codons. So if you look at this first one, TTT, it codes for phenylalanine, but also TTC will also code for phenylalanine. And what happens is certain organisms will have a carrier that matches this with this to bring this to the ribosome and make the protein. And other organisms will have this, meaning that. So, like, working between systems is really difficult because of this, but you can work in eukaryotic systems.
Can I just add a few things? There's some really interesting points about why we should use your eukaryotic systems and why we shouldn't. I think the easiest way to say why eukaryotic systems suck to work with is because we really rely on antibiotic resistance. If we want to put a piece of DNA in a cell, we always tag on an antibiotic resistance with it. So essentially, any of our experiments that don't work, the cells just die. And any experiments that do work, the cells live. With a eukaryotic cell, if it kills the cell you're working with, it'll kill you too. So it's very difficult with selection. You can't just use antibiotics. So it makes things a lot more difficult. Another thing is that they take a long time to grow, many of them. Where E. coli grows nice and quick, so you can have like a 24-hour experiment turnover time, where even working with yeast, which is a very quick growing eukaryotic cell, it even takes longer than E. coli to grow. So then just switching from kind of the next best choice, if you were going to eukaryotic systems, it basically cuts the time in half that you have to work. So you're now 2 months of time you have to work cuts down to 1. A good thing about using eukaryotic systems, though, is that, well, E. coli, have you guys heard of bacteriophages at all? Well, there's a lot of viruses that can infect bacterial cells. So if you have a system that you engineer into E. coli or any other bacterial cell, it can actually get sick. So if you actually start a company and start making these organisms in E. coli, your organism can get sick, just like us. E. coli can get sick, where if that's the time, then you would switch it over to yeast. So even if people do work in eukaryotic systems, usually they assemble all the DNA and do all the work in a prokaryotic system, so a simple bacterial system like E. coli. And then once they've got all that done and they can do it really quick in a bacterial or prokaryotic system, that's when they just move kind of everything they've worked on and they put it all into a eukaryotic system like yeast. So antibiotic resistance and time are the big things. Sorry, Anthony.
No, thanks. Is there any more questions? Yeah.
So like after the DNA is cut in the palindromes, you said that you need ligase to glue on the backbone again, is that right?
Mm-hmm.
Why do you need the backbone on it again, just to clarify? Is it going to attach or?
No, because
If the backbone is still broken, there's a chance that the DNA can come apart again, and then it won't be that nice circular form. So we just glue the backbone together so it maintains that circular form and it doesn't fall apart.
it's kind of like if you had Velcro and you're holding something together with Velcro versus if you were super glue it together. That helps.
Okay, that helps.
Yeah.
It's a bit of conflicting. Normally, if you end in ase, you're an enzyme, but if ligase puts something together, how does that work?
Sorry, what do you mean by?
A ligase is an enzyme, you're right. So it's a protein that works to recognize where those breaks in the backbone are, and then it'll glue them back together. So you're right, it is an enzyme.
Yeah. Because like even the restriction enzyme, they're enzymes. Everything, all the things that do function, most of them, from what we know from like science, is proteins, and they do that's why we went through gene expression earlier. So you have DNA which codes for a protein, and then you have a process that makes that protein, and the protein is what does the function.
Yeah.
Does that make sense?